Let us study transcription today. What is transcription? Just the conversion of DNA into messenger RNA. We'll see the details. Transcription is the first step of gene expression. What is gene expression? There is a gene, a certain portion of the gene codes for a certain protein. That protein has certain functions in the body. So from the DNA, a protein is made, or the function of DNA is to make some changes in the body. That is known as gene expression. So transcription is the first expression. So it involves formation of RNA molecule from the DNA. So that's first. So this is a broad overview. You see, there is a DNA. You can see the DNA, double-stranded DNA, which is a very lengthy material. The genetic information from the DNA to protein is happening through two steps. One is transcription, second is translation. You can see that at the end of transcription, the double-stranded DNA became single-stranded messenger RNA. Now, these messenger RNA will be taken out of the nucleus to the cytoplasm where the translation happens. So, translation involves what? From this messenger RNA, 3 nucleotides will be identified by certain amino acids, and that will be forming a chain. Okay, first 3 will be coding for one particular amino acid, next 3 will be coding for next amino acid. So a chain of amino acids will be formed, and that is known as a protein. So transcription is the first process in gene expression. In this, the DNA double strand will be unwound and the template strand will be used for synthesizing RNA from that particular DNA. Now this RNA or messenger RNA that is formed carries information to encode for a protein. That means the RNA or messenger RNA will be taken to the cytoplasm to form that particular protein. But why the DNA is converted to RNA? Why can't the DNA be taken out of the cytoplasm? This is because the DNA is having a heavy double-stranded structure. You can see DNA is present inside the nucleus. Okay, here inside the nucleus it is happening, and this is the outer layer of the outer envelope of the nucleus. In that there are certain pores. Okay, these are the pores, minute pores you can see. So this is how, if we elaborately draw one nuclear pore, you can see the central pore, but this is very, very small size. The double-stranded DNA is too large to pass through this particular site, through this particular pore, so it needs to be converted into a light or small single-stranded RNA molecule. Now the third thing is RNA polymerase. RNA polymerase is the main enzyme involved in the transcription. It uses the single-stranded DNA, as we told. Okay, when the transcription happens, the double-stranded DNA will be unbound. That particular area will be unbound to single-stranded DNA. That will be used as a template to form a complementary RNA strand. Now, the RNA polymerase binds to the promoter. I will show you that, and catalyzes the polymerization from 5' to 3' direction on the template strand. I will show you how it happens. Okay, this is the RNA polymerase. This surrounding area, the surrounding circle, you see the violet color thing. This is the RNA polymerase. Now it is attaching to the particular site of DNA where that particular gene need to be processed. Now you can see that this is the region which is going to be transcribed. RNA polymerase got attached. Now this strand which is not taking place in this particular process or gene action is known as coding strand. Okay. Now the opposite to the coding strand, you can see the template strand. But if you see, this is the coding strand and this is the coding strand. Okay, I'm zooming this particular area to here. You can see A, T, G, A, this is the coding strand, and the template strand, you can see the complement to coding strand, you can see T, A, C, like that. But if you see, you are making an RNA using this template strand, but finally what you are getting is a copy of the coding strand. You see this? Like a magic, right? You are using the template strand. Finally, in the RNA, you are getting the information that is present in the coding strand. Okay, A, G, G, A, C, C. Why I have skipped T? Because in the place of T, the only difference is that RNA will be having uracil in place of T. Okay, in place of thymine, RNA will be having uracil. So, as a complement to the adenine. Okay, in the case of coding strand it is T, and in the case of template strand it was A. So whenever the complementary to adenine need to be added, it is not thymine but uracil will be added. But finally you will be getting a copy to this particular coding strand. Fine. Now once the RNA, so RNA is moving, once the RNA polymerase is moving and reaching a terminator sequence, for example it's like UAG, UGA, UGG, like that, terminator sequence. Once it reaches there, the process terminates and the newly synthesized RNA strand is released. So this is how the process takes place. You can see that this is how, there is an area, okay, this from 3' to 5', this area, this is the area of the gene that need to be transcribed. So there is a site called promoter site. Normally it will be AUG, like that, the nucleotide base pair will be AUG, like that, the nucleotide order will be there. To that the RNA polymerase will be attached, and from, you can see this is the template strand and this is the RNA that is getting made. If the template strand is in the 3' to 5' direction, now the mRNA will be created in the opposite direction, 5' to 3' direction. So finally it reaches the terminator sites and it will be getting terminated. So what are the stages of transcription? First stage is the initiation, second stage is elongation, third stage is the termination. So this is how it moves. This is the RNA polymerase. This yellow thing has been moving from here to here to here to here to here to here to here. Finally it reached here. Now it identified a promoter, like a sequence like A, U, G, like that. Now it will be beginning the initiation. And what is this circle, white circle? It is known as sigma factor. Okay, we will be having a detailed video on initiation and all. In that time we will be seeing what is sigma factor associated with the promoter sequence, or which is associated with the initiation. Now at the initiation, RNA polymerase is attaching to the promoter sequence. Now it will be, in the next step is start elongation. Once the promoter initiation begins, now next step is elongation. So this is the template strand and this is the mRNA. This orange thing is the mRNA. You can see that to each nucleotide of the template strand, the complementary yellow, the RNA nucleotide base is getting added. Okay, fine. Finally, now this is continuing in this direction from 5' to 3' direction. It will be moving. Now you can see that at the end there is a terminator sequence and there is a protein called a raw protein associated with it. We'll see that in the termination specific video. Now you can see at this particular point, this particular RNA, mRNA will be forming a hairpin loop. I'll show you that in the later slides, and then it will be terminated. So initiation means what? You can take down the notes if you want by pausing the video. RNA polymerase attached to the DNA molecule and moves along the DNA strand until it recognizes a promoter sequence. So the promoter sequence are known as transcription start sites. I'll show you that. The DNA double helix then unwinds and all the bases of each DNA strands are exposed. So A, C, G, like that, everything will be T, everything will be displayed. Now this acts as a template for the new mRNA strand. You can see that this is the RNA polymerase. It is getting attached to this blue colored promoter region. In that specifically it is recognizing two recognition sites, transcription sites, two sites, that is minus 35 element and minus 10 element. By attaching to this particular site, it is getting stabilized in this particular promoter area. Now from here the transcription begins. So that process is known as elongation. So ribonucleotides, so adenine, uracil, like guanine, cytosine, etc. will be added to the template strand according to the copy that is present in the parent template strand. That will be added, okay, to the new mRNA. According to the template strand, ribonucleotides will be added to the new mRNA. So you can see that. So this is the transcription bubble, this particular unwound area. Now you can see like complementary to A, U is added, again A, U, G, C, T, A. So you understand that when T is there in the RNA, there is A, but when A is there, there is no T in RNA, so U will be added. So it is proceeding in the 5' to 3' direction. This is the template strand. According to the information that is present in the template strand, this complementary will be available in the mRNA. Fine. Finally, RNA polymerase encounters a terminator sequence like U, G, A, U, A, G, U, G, G, etc. And transcription stops and RNA polymerase releases the DNA template, which will be going to its normal state. So you can see RNA polymerase has finally reached the UGA, UGG or UAG site. Now it will be forming, this mRNA will be forming a hairpin loop like this. You can see it got transcribed like this, and this yellow portion and this yellow portion will start getting base paired because this brown area got hair loop, formed a hair loop. Now you can see, now this cannot escape the RNA polymerase. So it gets into trouble, so it cannot move further. So the transcription, the RNA polymerase cannot move further because it got blocked by this hairpin loop. Now transcription stops at this terminator sequence. Okay, fine. At this sequence it got stopped. Fine. Yeah. So after this, what? All the unbound area will be getting rebound. This is the termination site. Finally, a pre-RNA or pre-mRNA is released. So you can see something like polyadenylation signal. What is that? We'll see. So this is a summary. There is a DNA. In that, a small portion of the gene. I am zooming in. This is the gene. Now when the initiation happened, it is getting unwound, and in the elongation state, for each nucleotide base in the template strand, corresponding complementary RNA nucleotide base is getting added. Now this is the mRNA. Now it will be taken out of the cytoplasm where translation happens. Now UGG will be identified, will be the information for coding like tryptophan, okay, TRP amino acid. Then the UUU will be coding for another amino acid. Then GGC will be coding for glycine amino acid. So similarly, for each triplet of or codon of this particular mRNA, each corresponding amino acids will be added. That will be forming a chain which is known as protein. Fine. Now you can see this is the transcription. We have already discussed this thing. It is proceeding in the 5' to 3' direction. So finally, a process known as RNA processing will be happening. Okay, the RNA processing, I'll show you what is that. This is for stabilizing the RNA and for preventing it from degrading and for facilitating the easy export of RNA out of the nucleus. Three processes are there: capping, polyadenylation and splicing. Capping means what? At the end of the mRNA, a certain cap called methyl guanosine, etc. will be added. You can see this is the normal mRNA that got released from the transcription. Now to its end, at one end, at the 5' end, there is a 7-methyl guanosine got added. So this is known as cap. It prevents this particular RNA from getting degraded by the exonucleases, etc. Second thing is the second step of processing is polyadenylation. So you can see at the end of the mRNA which is created, you are adding multiple adenine. Okay, why it is done? It will be helping the mRNA to export out of the nucleus because it will be adding a recognition signal. It will be helping in recognizing the mRNA to the nucleopore. Okay, so that will be facilitating the easy removal of this mRNA out of the nucleus to the cytoplasm. Then it will be protecting the mRNA from exonuclease degradation. Exonucleases are enzymes which will be degrading nucleic acid. So that will be protected by this tail. Then if the tail length is more, that lengthening, that stabilizes the mRNA transcript. So that will be stabilizing the mRNA and preventing it from getting degraded. So final thing is the splicing. Splicing means what? In mRNA there are portions called coding region and non-coding region. Non-coding regions are called introns and coding regions are called exons. These non-coding regions are waste, that unwantedly added lengths. So that will be removed. That process is known as splicing. And the molecules that facilitates this splicing is known as small nuclear ribonucleoproteins. So that will be forming a complex of this protein and this particular mRNA known as spliceosome complex. Then finally that particular thing will be forming a loop and the exons will be added together and that will be released. This is transcription. This is the